So we are looking at enzymes today. Okay, very big part of biology. Lots of people tend to get quite stuck on enzymes, but I think that they're manageable when you break them down in certain ways and think of them with little hints and tips that are kind of useful for when it comes to thinking of enzymes. So the basics of what an enzyme is what we call a biological catalyst. Okay, when we think of biological, what we're really saying is it's a living thing, or it is and what's the best way of describing it? Biological, you're saying it's something that we have physically made that is technically alive in many ways. So biological, we're talking about it's realistically it's almost alive. A catalyst, a little bit more vague, is something that speeds up a reaction. Without itself being used up. Now the reason why I'm focusing more on the catalyst side of things is that we have catalysts in chemistry as well that aren't biological. So, you know, when we look at a chemical reaction in chemistry, it could be a metal that we use as a catalyst, and all a catalyst does is it pulls different molecules together and gets them to create a new product really quickly, or it does the opposite where it takes a molecule and it breaks it into two smaller pieces instead. All a catalyst does is it's trying to speed up the rate of your reaction. So it's about developing it and changing or creating that reaction quicker over time. So when we look at enzymes, they are living things that are doing this. We say living things very broadly here. Sorry, I'm just looking at the shine. Ugh. So they're things that have been created internally inside in the body that are going to be speeding up the reactions that we're having. That's what an enzyme is, okay? And a big key part is that whole idea of a catalyst. We'll come and revisit that later on. So at its core, that is what an enzyme is. Let me get it in different colors. Okay, so that's fine when we're thinking of catalysts, but let's focus a little bit more on how they are structured. So the structure of enzymes in the body, they're a little bit unique, okay? We have a long chain of amino acids. Okay, so these amino acids, we're going to find them randomly inside in our cell, inside in the cytoplasm, and some of you might be familiar with a TikTok I made a little while back where we were looking at we looked at tRNA, we had mRNA, we had ribosomes, we had DNA and RNA, all of that stuff. Well, we had one TikTok where we looked at what a ribosome actually does. And inside in a ribosome reads a messenger RNA, mRNA. Some of you might be familiar with that one. I know that's going to be a little bit more complicated if you're like junior cert or you're not doing your GCSEs, but if you're doing GCSEs upwards, this is the kind of the stuff you have to deal with in biology. But the ribosome is like reading the mRNA, so it's checking out all these codons, and it's telling this tRNA, go out and get me the amino acid that's going to match with this codon, this triple codon, this part of that mRNA that I'm reading. And the tRNA goes out to the cytoplasm, it finds the amino acid that suits it, pulls it back in, sticks it in, and that forms one part of this enzyme chain of amino acids, okay? So you get an amino acid, then another amino acid, you keep building it on, you get a big long chain of amino acids. And once you've made your long chain of amino acids, we can then fold it into a particular shape. That's the key with enzymes. They're not just like one long straight line of amino acids. The amino acids are folded into a particular shape. So it's a long chain of amino acids folded into a shape. And that shape is unique to a substrate. Now you might not have heard this word substrate before. Basically, it's just the chemicals inside in the body that your enzyme is going to be pulling in to actually have its little, like, I won't say reaction, but where it's going to be doing its little job to break them apart or pulling them back together. Okay, so that's the core details of what an enzyme is, and that word, catalyst. Right, I'll take questions. Any questions so far? Any questions? Someone probably want to screenshot that. That really bugs me that I can't get the light right. Hold on. Oh, I'll take that. Right, that'll have to do. Any questions? What level science is this? So this is probably, if you're doing your GCSEs, if you're doing your A levels or leaving cert, this is that kind of level at this point. What do enzymes do? So enzymes, the core basics of what an enzyme does is that it's used in, I won't say used, but it's what causes the chemical reactions inside in the body. So it's almost like the builder inside, like trying to take these different building blocks together, add them on and make a new product. It is essentially building something inside your body. But when we're saying building, we're talking about chemical reactions. Enzymes are proteins, right? Yes, enzymes are proteins. So when we talk about ribosomes, you quite often hear people saying, oh, ribosomes are making proteins. Enzymes are proteins. They are biological proteins, are biological catalysts, and proteins are involved in lots of the different reactions we have around our body. Yeah, great question. I don't understand any of this. Oh, well, it depends what age you are as well, so some of it might not be related to what you're learning just yet. What's an example of an enzyme? So great question. A good example, we'll get onto it properly later on, is it might be something like amylase that we produce in our salivary glands, because amylase is involved in breaking down starch. So that's where it looks at the starch molecule, goes, right, that's really big. Let's fit it in here and let's rip it apart into something smaller like maltose. Or when we look at maltose, then it might have maltase, which is again another enzyme. The enzyme will look at maltose and go, right, let's rip that into a smaller piece as well, and you get glucose. So there's a couple of them all over different areas in your body, but that's just ones inside in your mouth. We've got loads of different enzymes. What age is this for? Probably, what we're looking at, probably 14 upwards. Any other questions? Why do we need enzymes? So the reason that we look at enzymes as being really important, and the reason that like catalysts in general are really important reactions, is that they speed them up. So it's all about speeding the whole process up, because essentially when we have chemical reactions, you're just hoping that these different molecules are just going to bump into each other and that will cause a reaction. But if those molecules are moving really slowly, they don't have enough energy maybe, that when they do bump into each other, that they're going to have a reaction, that they're going to like end up causing this new product to be made. So you kind of have to get something in there that's going to speed it up. The way I would think about it is, imagine if you had a bath full of rubber ducks, okay? And you filled up the bath with water, you had rubber ducks all over the place, and they're all spread out, okay? And if you just stood there and you were like, right, I can only take the rubber ducks out when two of them bump into each other. Well, if you're just standing there waiting, you know, you could be waiting forever for two ducks to bounce into each other. And you're just like, great, this is taking forever. So to influence it, you come along, grab two of the ducks, and you just bump them into each other, and you go, oh great, I can take those out now. That's a bit like what an enzyme is doing. It's just taking two different amino acids or whatever else is in, oh sorry, not amino acids, but it's taking two different pieces of something and just pulling them together and making something new, or breaking it in half. Okay, you're kind of, there's two different groups of enzymes. We look at the ones that are catabolic, which is about taking things and breaking them down, and then anabolic, which is making them bigger afterwards. So there's kind of two variations of those enzymes, but we'll get into it a little bit more later on. Are you right? Ooh, someone said that. Are you writing the definition of a substrate? I will get to what a substrate is in a second. I was just kind of giving the general overview of what an enzyme is and a catalyst is, but I will definitely go with it. Sorry, disconnected, but I joined again. Cool. Is this GCSE or A level? It's mostly GCSEs. There will be parts that are probably going to push towards the A level though. What if it gets slow? You might be talking about the different things that are affecting enzyme activity rate, which we'll get to in a second as well. Let's mute this person. I don't care about Nicki Minaj. Sorry. What's the point of that? Mute. Sorry. I hate that. I hate when people just come in and just start absolutely just ramming the comments in that are absolutely useless. How do you do percentage questions? Do you know what? Ask me that question over on my Instagram because it's not related to what we're doing right now, but we'll get there. Are you Irish? Yes, but I teach in the UK. Right, let's move on. So, an enzyme, like we said, biological catalyst. Its structure, it's got this long chain of amino acids. It's got a fold that's going to be unique. Now I've realized why I can't have the light facing that way, because you can't see the colors, or you can't see the board properly. So it's folded into a unique shape that's specific to a substrate. Now a substrate is just going to be a chemical or a substance inside the body that is going to be fitting into a particular part of this enzyme, so it can be broken apart. We'll talk about the method of breaking apart that now. Where's the wipe I need? Sorry, I need to get a wipe for the board. It'd be handy if I had one of those. So how enzymes work is this method that we call the lock and key method. Some of you will probably have heard of that from school, where essentially we find this unique shape of an enzyme, and we find the substrate, the substance that's going to be fitting into it. And we're going to be breaking it, or we're going to be adding them together. So I'll show you the two different types. So we've got catabolic enzymes, and we've got anabolic enzymes. Happy? Right, let's go with, do I have an example for each? I think I've only got an example for one. Anyway, doesn't matter too much. So let's say that this is our enzyme. Okay, it's got this random shape. Okay, so this here is going to be the area that substrate, the substance itself, is going to fit into. So that's our enzyme. Okay, it's got this unique shape. You're going to have a substrate that fits it. Okay, so it's got that unique shape. If there was another one out here randomly that had like this shape, that doesn't fit in here. So you can see that one wouldn't be suitable, whereas this one is suitable. So this is going to end up going into this space, which causes it to look a bit like this. So we can see it's fitting into it at this point. So it's after joining in, and this little area here, where the enzyme and the, of course this isn't going to write for me now, is it? Why is this pen not working? So this area in between where we've got the substrate and we've got the enzyme, where that actual reaction is happening, we call that the active site. But this whole thing where we have the enzyme and the substrate all together, we call that the enzyme-substrate complex. But that just basically means you put your substrate into the little gap, it fits in there, like a key fits into a lock. That's why it's called the lock and key method. Okay? So it's in there at the moment, it's got this little reaction that's happening just here, and the next part that happens is that enzyme will break it into two smaller pieces. So the enzyme, still here with the exact same shape, is going to break that substrate into two smaller products. Actually, I need to make it look a little bit more like it fits the shape. Okay, so it's after breaking it into two smaller products. So that's catabolic. Catabolic is where breaking it, we're taking from big to small. Okay, so catabolic, we're going big to small pieces. Okay, now this is where, like, if you're a bodybuilder, they always talk about being catabolic, and after you've like done a workout, your body is breaking down your muscle tissues. That's what's happening. You're going catabolic, your body starts to break down the muscle tissues into smaller pieces. Okay, so that's a catabolic reaction or a catabolic enzyme. So again, little words that are important, we call this the lock and key. Okay, so this is a lock and key structure. You've got the enzyme acting as the lock. The key would be the enzyme, or would be the substrate. The substrate goes into the gap. It breaks it in two, and it's after making two new substances. So it's gone from a big substance down to a smaller substance. Okay, that's the whole process here that we have for catabolic. Now we go the opposite with anabolic. So this time we might have another one where the enzyme, I'll put it in the almost exact same shape just for the fun of it. Okay, the enzyme again, it sees that it's got these little pieces outside. I'll put them all over the place so that they don't look like they're already together. Okay, it's got these little pieces here, so it says, right, let's add them in. Okay, let's push them into that enzyme. The enzyme itself then, again, same situation as before. It's going to now have these two pieces inside here. Going to act like it's split down the middle. We still have the active sites where that reaction is occurring. Okay, so we still have our active site. And then finally, that enzyme is going to release it all together. So instead of it being two separate pieces, it's going to release it as one giant piece. I know it's way bigger now, but I'm just exaggerating a little piece. Actually, let's even it because I won't be happy when you look back at it in the video. Okay, so you can see here, it's the reverse situation. This one was taking one piece, putting it in, active site is reacting with it, or is causing the reaction to break it in two, and then it splits into two new pieces. So we've gone from having one piece to two pieces, whereas here in the anabolic situation, we're going from two smaller pieces to a big piece at the end. So we're going the exact reverse of catabolic. So two unique pieces come in, they're going to be reacting, or they're going to have the reaction in here at the active site, and then it's going to release it as one big piece. I'll take questions. Questions, anyone? Is this maths or science? This is science. What is this? This right here is what enzymes are doing. So inside in the body, your enzymes are going to be reacting. Some of them are breaking it down. So you can see this first one, it's going to be taking that one substrate, it's going to be putting it into the active site, and it's having its reaction, breaking it into two smaller pieces. Whereas down here, we've got these two smaller pieces. They're going into the active site. They're going to, ooh, I've gone too far now with my finger. It's reacting, or it's having its reaction together, and then it's making one new piece here. So you've got two completely different systems, but they're doing the exact same job. They're taking something and they're making something new afterwards. One is going to be making two new things, and this one is going to be making one new thing. Can you explain negative? Oh my God, what does this occur? Where does this occur in the body? So this is happening all over the place. So I was saying earlier at the start, this can start somewhere as basic as up in your mouth, where you're going to have enzymes that are like taken out of your salivary glands. So when you produce saliva, you've got these enzymes in there called amylase. And as soon as you eat food, it's going to start looking for starch molecules. It's going to go, right, I found some starch. Starch is a big long chain of these glucose molecules. So what I'm going to do is I'm going to intentionally try and break those into two smaller pieces together so that we end up making maltose. And you get your maltose then, and there'll be another enzyme called maltase, which is then going to break the maltase down into smaller pieces into glucose. So it's constantly breaking them into smaller pieces. Just like what's happening here in your catabolic enzymes. They're going to be taking one big thing, breaking it into something smaller. Whereas in anabolic, so So this is where you hear the word anabolic steroids from. If we're looking at anabolic steroids, it's about taking the building blocks of the proteins to build new muscle tissues. And they're constantly adding muscle on. So it's more and more and more, building up, 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 getting bigger, bigger, bigger. Whereas with these ones, it's about breaking it down into smaller, smaller, smaller. So they're two complete opposite situations. Questions? Let's say we did this for junior cert. Yeah, some of it is the basics for junior cert. More of it starts to push on towards the leaving cert as well. We'll talk about it a little bit more. Is that not the induced fit model? We will be talking about induced fit a little bit more because it's like a variation of this, but it's very, very close. A-level or GCSE? GCSE predominantly. Can I give an example of a substrate? So we had starch there a while ago. Starch is a good one. Maltose. If you were looking at something like hydrogen peroxide, H2O2, that breaks down into something like water and oxygen gas. That would be, again, you'd have something like Catalase would break that down into those two smaller pieces. Is the breaking down continuous? Same with the building up. To a degree, like it depends on, like you keep breaking things down if you have things that are able to be broken down. So it's like if you keep eating more and more starch, so every time you eat, if you have some starchy kind of foods there, then you're giving your amylase in the saliva a job to do. But if there's no starch there, then it can't operate. That's the key thing to it. So like when bodybuilders, right, this is where the kind of the whole catabolic anabolic situation comes from. If you go to the gym and you have a workout and you start breaking down muscle tissues, there's enzymes in your bodies that are physically breaking them down. Now that's important because you're going to have those tears and a certain breaking down anyway for you to be able to build more muscle. But if you don't have the protein that you need afterwards to build more muscle, then it's going to try and find that energy to build or repair muscle from somewhere else. And that's where people like start getting smaller over time as well. So there's a certain degree of it that you have to like look at where you're going to get your building blocks for in the future. That's the key part of it. It's a bit more complicated than what I'm explaining, but I'm trying to keep it as basic as possible. But that's why they are talking about, you know, you have to have protein after you go for a gym session because it's going to be giving your body a place to find the protein it needs rather than trying to take it from somewhere else that already exists. Oh man, there was loads of questions that popped up. What's an example of a catalyst? So in the body we had amylase, we have catalase. They're biological ones. There's loads of enzymes in the body that are biological catalysts. If you look outside of, so we go to chemistry, most metals and transition metals in particular will also be a catalyst, but the key difference is enzymes are biological ones. So we're going to talk about denaturing in a minute and kind of the responsibility that plays. But I'm going to put it back to that one that someone said a while ago about the induced fit model. So this is fine and straightforward when we talk about it as if the enzyme's got a particular shape and then something just slots into it, the lock and key method. But there is a separate one called the induced fit, and it's really similar, and it's a bit like when you sit into a beanbag, okay? So if you've sat on a beanbag before, you'll know the beanbag has a particular shape before you get into it, but once you sit into it, that beanbag kind of curves in around you and it like induces a fit around you. Enzymes apparently act very similarly now as well. That's the belief anyway, that these enzymes already have a certain shape, but when they find its unique substrate, the substrate will go in there and then the enzyme kind of closes in around it. It has its reaction at the active site and then it breaks it up and moves it elsewhere. Okay, so that's the kind of the other way that we can look at it. The lock and key method almost sounds like, yep, it's as unique, that's it. It doesn't change its shape in any way, and then the substrate comes in, we just break it or we make it there. And that's it. Whereas the juice suggests that the enzymes can just about bend a little bit and create a little new shape around it. Anything else? Any other questions? Quickly while I'm having a look. Anabolic and catabolic hormones, yeah, there's loads of them as well. So like when we're looking at your hormones, they're built up of all of these enzymes as well. So yeah, great examples. Anything else? Oh, someone says, does this happen with what? Does this happen in the stomach? Yeah, like this is literally happening all over the body. So there's going to be different ones depending on where in the body you're reacting. Someone said there a while ago, for example, like hormones. So like When we think about your salivary glands are going to be producing certain ones, your pancreas is going to be producing certain ones, your liver might produce ones, your stomach, etc. All of the different parts of your body are going to have different enzymes doing different jobs. So that's why they have to be a bit unique. They're doing certain jobs in certain places that you might not necessarily need anywhere else. Okay, so that's kind of a really important one. What did I have down that was going to be linked to this? Okay, yeah, so some people are going to be a little bit confused about why we say enzymes don't get used up in the reaction. So the way that I like to think about it, and you can take this if you want or feel free to use it in this example, I always think of it as like someone who is stirring the pot and breaking up relationships, and someone who is playing the matchmaker and coming back and making something together. So I always think about it as that person in a year group who is like always trying to start a pot, like loves a bit of drama, breaks couples up, and this is a bit like a teacher trying to rearrange their seating plans to put couples together. So you can imagine it. The enzyme here is a person, they see a happy couple together and they go, I don't like the fact that they're so happy here. I'm going to just get involved, be that little voice in the ear, send a couple of messages or a snap and like just stir the pot to say that, you know, oh, you're really sure you want to be with that person, ends up breaking them up, okay? That's what a catabolic enzyme is a bit like. It's that person that just loves a bit of drama, okay? The anabolic enzymes are like the teachers in classrooms. Not all teachers, but some teachers, okay? Because we all know some teachers will do it, or at least it's a shock. Some people are surprised that some teachers will do it, but it's like we see these two students in the class and we go, do you know what? They'd be a lovely couple together now. I'd love to see the two of them happy. So what I'm going to do is I'm going to sit them next to each other at the front of the class here, and we'll just give them a bit of time, see if that, you know, if a little spark or a little chemical or a little bit of chemistry forms, and hopefully we'll end up making a new couple together, okay? So it's like a nice unique way of thinking about it. This one is the person who loves to stir the pot and a bit of drama, break everyone up, and this one is the one that is playing matchmaker. In both scenarios, this person isn't ending up like connected or stuck to anyone in this one. And likewise, the teacher here is just playing matchmaker. They're going to still end up being the lonely teacher who has no girlfriend or boyfriend. All right, they're just going to be the person who likes pairing up the students, right? That's how it works. Easy way of thinking what enzymes do. They're just catalysts that bring them together. These two might have been continuously happy for the rest of their lives if this enzyme didn't come along and break them up. Likewise, these two students might have never had the opportunity to sit next to each other and actually start to get to know each other if it wasn't for the teacher pairing them up. So catalysts are things that are speeding up the odds of a reaction happening. That's all it is. That's all catalysts are. Right, questions? Sorry, I'm late. No worries. Anyone? Can I do reproduction plants next week? Reproduction plants isn't scheduled in for me for a few weeks' time. I'll be back to chemistry and physics next. Hello. How are we? Can I screenshot? Yeah, screenshot away if you want. Go for it. Is this higher foundation? Higher free science class. Woo! Does this happen in animals too? Yes. So this happens in any living organism. So you're talking about animals, plants, fungi. You're going to have them pretty much everywhere, okay? There's going to be some sort of enzymes used to break down things and to make new things. Wait, what? I just joined this. Sorry. Yeah, we're talking about enzymes. Sorry, I'm late. Wi-Fi problems. No worries. Is this good for the enzymes? Is what good for the enzymes? Like over time, the enzymes don't get as good at it anymore. They kind of like, they get tired and slower at actually changing and making new products. But generally, the enzymes don't change. They're the exact same as they were beforehand because they're not actually being used and involved in being part of the new product. So they're kind of kept separate afterwards. They're just like, that's why I like to say the matchmaker one, because they aren't in the new relationship, but they helped become, or they helped make that relationship happen. So that's the easier way of thinking about it. Now, what was I going to say with that? Now, there is one issue, or what we have a concern about with the enzymes and the active site, is that there are a couple of things that can end up affecting the enzyme and its activity, or the rate of its reaction. So there's three things that we can look at that would affect how well it works. So let's get rid of catabolic, because now we're just talking about what are we talking about instead? What will I call it? let's call it effects on rate. So there are three things that can affect the rate of activity of these enzymes. So the first thing that could affect it is the pH. Now the pH will only really affect biological catalysts. You don't tend to find it having the same effect as on the enzymes, on the catalyst as you do when it's in chemistry, because in chemistry the pH doesn't necessarily affect the rate of reaction, whereas the biological part, it's going to end up denaturing it. I'll put that word in a minute. But the pH is one, temperature is the second one, so how hot the reaction is, and then the third one is concentration. Basically, how much of the substrate we have. Okay? Now, overall, these things, the first two in particular, can affect the shape of your enzyme and cause it to denature. And when we talk about denaturing, all we're saying is we're altering the shape of the enzyme and stopping it from being so effective. Like if you're going to be changing the shape of your enzyme, your substrate might not necessarily fit in there properly anymore, which is going to end up meaning that it's not going to fit into the shape. We can't break it down or we can't make new things. So it stops being effective. It's a real big issue. If you want to be working as effectively as possible, you need to make sure you're at the right pH and that you're at the right temperature. So let's talk about what kind of graph we're going to end up seeing if we start changing these things. So we'll start by looking at pH, because it's the most straightforward one, most linked to the biology part of the side of this course and what catalysts are. Again, I should be saying, for anyone who is here who is new or has any questions that I'm not answering that for some reason, maybe I'm not seeing your question, maybe it's not linked to this topic, over my Instagram, I open up my story so that you can ask me questions on my Instagram and I'll get back to you over the next 24 hours with an answer, because sometimes there's questions you have that just aren't linked to what I'm teaching, so it gives you a nice chance to ask me there. And likewise, my YouTube, I've got loads of stuff up there at the moment. A lot of these live lessons are going to go up there as well, so you can look back at some of the old ones. I think I have 4 or 5 of these long ones saved now, so I'll probably put them over the next couple of weeks. Over the Easter break, you can look back over it if you want it as like background music, well, not music, but background noise, so that you have something that you can use as a baseline of knowledge. So in terms of our graph for pH, we'll call this side the activity level, or the activity rate. And down here is going to be pH. So this is going to be 0, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 14. Beautiful. So we can see that's going to be 1, 4. I know I'm just doing this spread out now. 7. Okay, so that's our pH scale, right? In terms of activity, there's going to be some of them that are going to be more active at certain pHs than others. So if we take something like, which one deals with Pepsin, there we go. So Pepsin is a good example of one that acts in the stomach. So Pepsin, in terms of its activity, it doesn't like to work very well until it hits a pH of around 2. Okay, so when it reaches pH of 2, it's at its most active point. And by the time it passes down and gets to like pH 4, it stops becoming active again. Okay, so it stops working as effectively. So this might be something like Pepsin. Okay, that's an enzyme that's in our body, inside in our stomach. So that's why we know, in terms of our stomach pH, it's somewhere between 2 and 3, okay? Pepsin works best at pH 2. It's what we call its optimum pH. Now another one that's in our body that we have, which one is it? Trypsin. Trypsin, again, something very similar. Trypsin works best at pH 8, and either side of it, it doesn't tend to be very active. Okay? So that's where we see two very different pHs, but two enzymes that work differently at them. So that's trypsin. And again, pH of 8 is trypsin's optimum pH. Anywhere outside of that point, so particularly here, there, here, here, and here, this is where the enzyme starts to denature. So if something denatures, the enzyme stops working, it changes shape, doesn't work as effectively anymore. So that's how it works when we look at pH. Okay, pretty straightforward. You can see outside of its optimum, it sharply decreases in terms of its rate of reaction and its rate of activity, and that's where you quickly see it denature into something else. If we put trypsin into a pH of 2, it's going to denature immediately, stop working. If we did the same with Pepsin, we put it into a pH of 8, it's not going to work, it's going to denature, it's not going to do us any benefit. So we have to make sure wherever we're working in the body that the pH suits the enzyme. The stomach, in particular, we see pH 2, way different to what our pH is all around the rest of our body. So Pepsin is unique in that scenario. Questions? Any questions? Where's trypsin found? Very good question. I can't remember. I feel like that's inside in your intestines, but I could be very wrong. I just remember that that pepsin is inside in your stomach. Trypsin pH 8. I feel like it's the intestines, but I could be wrong. Someone says an inhibitor can change the shape of the enzyme too and make it useless to a substrate. Yeah, completely agree. So again, inhibitors, same, act the same. Can you do radiation chemistry? I will, but I can't remember when I've got that lined up. I think I'm doing the maths part of chemistry next week. Chili's doing GCSEs, like they know that they're getting lied to. Shh, don't tell them yet, okay? Don't tell them that everything is lies up until now. This is where it freaks them out. So you start going into like the leaving certain A-level stuff and they start going, that's not what you told us in GCSEs. That's not anything like it at all. Can you do chemical bonding soon? I've already done it, Maddie, and I've actually put it already up on my YouTube channel. Go check it out. They're already there. What affects our body's pH? Loads of different things. So, like, generally, depending on what substances you're having yourself, that can affect it. That's why, like, if you think of your stomach acid as being pH 2 or pH 3, if you take something like Alka-Seltzer or you take like Gaviscon or something that's going to, like if you've got heartburn, what you're dealing with there is that your acid in your stomach is too acidic or it's going up your esophagus and it's burning it a little bit. So you have to take something that will neutralize it and change the pH back to 7. So you're looking at getting it back to like the pH 7. So you have to give it something that's slightly alkali. You're balancing it all out. Generally around our body, I think our body is mostly pH 7. So you have to just kind of have a fine balance of what you're taking in and using. What's today's topic? We were looking at enzymes and we were just talking about the effect of pH on the activity of certain ones. What about kidneys? Can you talk about those? I will, but not today because it's not linked. But ask me if you got a particular question. Ask me over my Instagram. Any other questions? I'm just seeing people answering questions, which is quite nice. An bhfuil cead go dogairí an lethir. Sean, tá cead. Go on. Love your accent. Cheers, thank you very much. I just saw someone ask, someone I think I recognised, would rather watch them. Well, what I watched this in my PD. Thanks, Cursor. Well, what I love about that as well is I know that you don't need any of this for your school at the moment, but I know that you've got messages over on my Instagram. I need to get back to you at some stage. I just haven't had a chance, to be honest. Can you go over why pH denatures the enzyme? So yeah, I'll quickly do it and then I'll move on to the next one. So in terms of the pH, what the pH is going to do is going to change the shape of the enzyme. So we were talking a while ago about this induced fit model, or we had the lock and key mechanism where a certain substrate would fit into a certain shape of the enzyme. Now if we change the shape of the enzyme, so it's like changing the shape of the lock, then the key that goes into it, the substrate, doesn't fit it anymore. So the pH is changing that shape, causing the substrate not to be able to get into it. So that's kind of the key to this whole lock and key thing. Right, that is the effect of pH. Let's now move on to, let's look at temperature. So temperature is very similar for a good chunk of this, but it changes when we look at the start point. So let's rub most of this out. So let's say now we're dealing with temperature. Let me find a nice one in the middle. We'll say 35. I'm doing this quite close, but you'll get what I mean once I draw it out anyway. So our body kind of works well around 35-ish, 35 to 37 degrees is where our body temperature works very well at. So we try and keep the temperature of our body around that because our enzymes like to work well around 35 to 37 degrees. So our activity level for enzymes are always going to be climbing towards that. Now, before that temperature, the enzymes will actually still work, but they're just going to be very, very low in terms of the rate of reactive, in terms of their activity. So they're not going to be very active until it reaches the 35 to 37 kind of area. And even going down 1 or 2 degrees is actually quite dangerous in the body. So 37 is where it loves to be. So 35 even is a bit of a stretch in terms of enzyme activity. But the problem that we have is as soon as we go over that kind of 37 mark, the activity level drops rapidly and it'll actually completely stop at a certain temperature. Now I'm saying it's just after 40 here. It's probably a lot sooner, to be honest, but my graph is quite small. But the key here and the key difference between that and the pH one is that enzymes, when they're colder, will still work. They just work very, very slowly. But go above it, it stops working completely. They want to be at their optimum temperature. So the optimum temperature okay, that's going to be the optimum temperature, and as soon as you go above your optimum temperature, you're going to be looking at denaturing again. But below it doesn't seem to be as catastrophic as what you see with the pHs. With the pHs, you get denaturing really on both sides. This one, you just don't see the activity being as effective. It's more that it slowed it down. It's a bit like how a freezer works. A freezer works on the same kind of principle that you're slowing down the effect of the bacteria. You're not essentially killing it. You're just extending the life of the food by having the bacteria and viruses, whatever, in there, not being able to work as quickly or as effectively. But as soon as you take them out and you put them back to its normal room temperature, and in this case, the normal temperature of the body, you're giving the enzyme a chance to work at its preferred rate again. Okay, so that's the key when we're looking at temperature. Any questions? Any questions about that? Oh, we're getting silly people in. Truth or dare? Sorry, I'm late. A dog stole my pen. That's a pretty talented dog. Someone's asking, we're just getting towards the end of enzymes now. What time have we got? Yeah, definitely getting towards the enzymes. Are you fluent in Irish? I speak a bit like Nil sé liafa, tá sé, beagán lufa in Orira. Moles, please. Moles I'll actually be covering next week. Oh, let's get rid of this person. Just acting the Egypt. Sorry, Johnny. Right, so that's what temperature is doing. It's just going to be denaturing it after it gets past the optimum. That's kind of the basics of it, nothing too major. Let's go to the final one, which is concentration. And then we'll probably be ending it there, just because I'm looking at the time. I'll get a brand new one for this one. So again, we're looking at activity rate. And this time we're looking at the concentration of your substrate. So we're increasing our concentration of substrate. So generally, the substrate, I should have write that as well. Generally, if you've got no substrate, you're going to have no activity. Okay. Now, the more that you increase your amount of concentration of your substrate, so the more stuff that you have available to react, the greater your rate of activity. Okay, so it kind of is in a linear point. So it's like if I've got two kids in the class and I'm like, well, let's pair them up. We can sit them next to each other. Hopefully we'll make a couple. Great, we're going to make one couple. If I've got four kids in the class, go, oh, well, maybe I'll be able to pair these two with this one with this one and this one with this one. I can have two couples. And I keep pairing and pairing and pairing. But there's going to get to a stage where I'm going, there's too many people that I can try and pair here together. I can only manage trying to do so much. So you get to a stage where you end up just plateauing. Okay, so that's where you reach your maximum activity rate. And it's only because the enzymes are too busy trying to already pair enough up. So you can have as much as you want of the substrate, but I can only work as fast as this. This is the point that I can only work to, okay? So it's the most it can, most the enzyme can work at. So it's the highest activity rate of the enzyme. If it's lower, fine. I mean, I could be, I could be looking at this being the first activity level. This would be the second. This would be my third. But you can see here, I'm going to be struggling after that. So this is like my absolute maximum. I can't actually do any more reactions at that time because I'm just full up at that stage. I just physically can't create any more. So that's what happens with enzymes. If you give more substrate to an enzyme, it will work as quickly as possible, but it could still be stacking up behind it, and you're kind of going, oh, there's so much of it there, but I just can't physically get to it. I've got to work at this rate. But if it's like quiet and there's not a lot that's there, it'll just do the small amount it has to do. It's a bit like people who are working in shops. They're going to have times where they're really busy and times where they're really quiet. When it's really quiet, they'll just do the bit of work that they have. They're going to be left over just waiting. But if there's loads of work going on, they're trying to work as fast as possible. If there's like extra customers that they can't reach, then they're not going to be able to help them until they've dealt with everyone else that was before them. Enzymes are doing the exact same thing. It's like it's got a little queue of who it has to help first and how it has to have its reactions, and then after that it'll get the new ones in for theirs. So that's kind of the guts of what the three things are that affect the rate of the enzyme activity. Right, questions? Oh, thank you very much for the TikToks. Is the graph supposed to be curvy? Well, it's supposed to go up like this and then across like that. That's only for concentration. I did have curvy graphs a while ago for pH and for temperature, where they curved up and back down again. So that would be your curvy ones. But for concentration, it's just up and it plateaus. Okay, so if in terms of increase, my concentration just goes up and plateaus for as long as possible, because that shows me this is my maximum rate of activity. But the other two, yeah, they curve. Any other questions? What are we doing right now? I'm literally just finishing off enzymes, so I'm going to take any questions. Got to go. Bye. Perfect timing. I'm listening to what you're doing while I'm doing my nails. Oh, love it. Do you do this every day or just Wednesday? Every Wednesday at the moment. I might increase it in the future. I don't really know, to be honest. I know it sounds awful. I wasn't actually in the mood for it today. I've been kind of sore and a little bit injured the last couple of days, and I was like just not in the mood. But yeah, I said I'll just do it. Hopefully over Easter I'll get around to maybe doing 2 a week. We'll have to wait and see. Do you teach maths? I do not. There are other maths teachers on here that do it. There's other English teachers. There's like, you ask me what subject and I'll find you a teacher. Just commented on one of my TikToks. So put into my comment section going, I'm looking for a teacher in whatever subject, and I'll tag one for you, and then you'll be able to go find them there. Am I doing live lessons over Easter? Yeah, I think I will. Can you do a Zoom instead of it? I probably won't do a Zoom because they're pretty hard to manage, but I might do something on YouTube where I can actually use a screen, so I'll be able to do proper presentations, and we'll go at it from there. What is a substrate? So a substrate is basically the substance that the enzyme wants to break down. That's what a substrate is. I hate school. You're probably not the only one. But look, if you enjoy TikTok, follow this page and I'll just teach you a little piece as we go along. Can you do it through Irish? I wish I was skilled enough to do a lot of this through Irish, but I don't have the skills, unfortunately, which is kind of sad. It actually makes me a little bit upset that my skills aren't there for speaking Irish. Yeah, someone said substrate is key, enzyme is a lock. Perfect, exactly. So the substrate fits inside the enzyme, the key fits inside the lock. Perfect. Oh, someone's like, no, carbohydrates and stuff. I've missed that question. Any other questions quickly? That question about the competitive and non-competitive inhibition, will you do me a favor? Put that on my Instagram story. I'll put a little question box up there because I'll give you a lot more detail for it there, and I'll give you diagrams and stuff as well. So just put it over there and I'll give you a full answer over the next 24 hours. Do you know any Irish teachers? I do over on Instagram, but there's actually not many of them that have come onto TikTok yet, which is a shame because I know that there is definitely a need for it. There are a couple of people who like do a lot of teaching in terms of like encouraging Irish, but they're not Irish teachers, if that makes sense. Where is the carbohydrate, protein, lipids produced? In terms of, it's a bit of a funny phrase question there. In terms of your carbohydrates, generally we're not producing them. You're looking at plants that produce carbohydrates. So maybe that's what you're thinking about. The question's a bit funky, but yeah, plants tend to make the carbohydrates for us. So the likes of glucose and starch and all of those different ones that are different types of your carbohydrates. Plants tend to make them, and then we take in the plants and we might break them down into different parts. So you might take something like starch, use an enzyme like amylase, break it down into maltose, then you take an enzyme like maltase, breaks that down into your glucose molecules, and then it goes from there, that kind of thing. Then you might be looking at your, I'm trying to just kind of figure out what kind of question you might be basing that around. Proteins, then you're looking at like the same thing. The proteins are made up of different peptides and your amino acids. So it might be a case you're taking in the proteins and you're using certain enzymes to break those down, like protease, which breaks them into the smaller chunks, the amino acids that can then be rebumped into making a new protein over time. So I think, I'm not quite sure what your question was about whether we make them or if they're being produced by something else. Generally, we're looking at carbohydrates being produced by plants, and then our enzymes refigure amino acids to make proteins. I hope that's kind of answered your question. Yeah, but where? That's like literally anywhere. It just depends on what you're trying to make. So in terms of like, if you're looking at making proteins and making enzymes, you're looking at your ribosomes. Your ribosomes are going to be making your proteins, but the carbohydrates are being made in your plants, if that makes sense. Luke, anytime. Thank you very much. You're welcome. You're so helpful. Keep doing what you're doing. Thanks. Look, Owen, just doing my best, Owen. Sometimes not, like today I'm not in the mood for it, but look, you know, I'll try and keep doing them every week, so at least you guys will get something beneficial out of it. Hydrogen bonds, yeah, they're just a type of intermolecular bond where the hydrogens have a little positive kind of charge to it, so they kind of push and kind of want to connect towards slightly negative stuff. So can you explain hydrogen bonds? Yeah, just going through it there. So the hydrogen has a slightly positive part, then it'll be attracted towards something that's slightly negative, and that will just kind of pull them close-ish together without actually being fully attached to each other. Legend, thank you. Can we learn something that you would learn in sixth year, sixth class? I need a little bit more details. There's a big difference there. Do you do ag science? I can, Dave, but I haven't taught it in a couple of years because it wasn't needed over in the UK. I will probably get around to it at some stage, but not in these live lessons. If I do it, I'll probably do it over on YouTube because I figured that's probably the place where I can actually put a full series together for you guys. Sixth class, sorry. I will try and do some experiments maybe at some stage. I know that like lots of sixth class and first year kids want to be doing some experiments at home, so I might see if I can make one or two over Easter, because then you'll be able to like do it while you're on your break and just enjoy it. Any other final questions? Because I'm going to finish up now. I need to sit down. My leg is killing me. Well, lad. Well, Harry, how are we? Any final questions before I go? Hi, Sean. Well, sir, can you do physics? I actually will be doing physics next week with chemistry, so I'll probably do, you know what, because it's Easter, I don't know what I actually even do a piece of each, so I might do half an hour each day of biochem and phys, give them their unique days. We'll wait and see how that works. Still need to decide. Can you do the periodic table?